Good morning, everyone. Welcome to our new discussion. So today we will talk about cell modification and cell cycle. As a review about cell, what is a cell? A cell is the basic structural and functional unit of all living organisms. What else? Cells are the smallest entities that can carry out the processes necessary for life. Also, cells are referred as the building blocks of life. Cells can vary in size, it can vary in shape, and it also varies in function. But they all share common features. And what is that? They have cell membrane and genetic material in the form of DNA and RNA, and the ability to carry out metabolism and to reproduce. So as a review, there are two main categories of cells. They are the prokaryotic and the eukaryotic cells. So when we say prokaryotic, what does it mean? It is the simplest form of cell. They are simple cell that lack true nucleus and membrane-bound organelles. Example of prokaryotic cells are the bacteria and also the archaea or ancient cells. Eukaryotic cells, they are complex and have true nucleus. They also have membrane-bound organelles. So what are those membrane-bound organelles? We have the mitochondria, the endoplasmic reticulum, Golgi apparatus, and the lysosomes. So eukaryotic cells are also found not only in animals but also in plants, fungi, and protists. Okay? So, when we say cell modification, what does it mean? Modification. The word modification refers to the act or process of making changes. Modification, modify. Murag motor bag, di ba? Motor ninyo, mga boys. You would like to modify or change the color or the aesthetics of your motorcycle. No. Tangag mga sticker, mga suga-suga. Unsa pay nindot i-modify? Mga sakyanan, i-modify. Okay? So, typically, modification means to improve, adapt, or adjust into a specific purpose or requirement. So, it involves what? Changes, alterations, adjustments, or variations made to an existing entity or concept to suit a particular context or need. That is the general definition of modification. How about in biology? In biology, modification refers to genetic alterations, changes in traits, or adaptations that organisms undergo to survive and reproduce in their environment. It can also refer to the changes made in the laboratory setting to the genes, organisms, or biological systems to study their function and behavior. So let us discover now what are the modifications that the cell made in order to function. Okay? So, in our learning competency, we will describe some modifications or some cell modifications. What is the purpose? That lead to adaptation. Why we need to adapt? Why the cells need to adapt? To carry out specialized functions. Examples are the microvilli and the root hair. So what is a microvilli? Microvilli are tiny finger-like projections that extend from the surface of the cells, particularly in the epithelial lining of the cell intestines and kidney tubes. These structures significantly increase the cell's surface area, which is essential for absorption and secretion processes. So the cells need to have microvilli in order to increase the surface area of the cell. Why we need to increase the surface area? Of course, to absorb more nutrients and for secretion processes. Okay? So that is the purpose or the function of the microvilli. So what is the structure? Ano sa itsura sa microvilli? You can see here in a microscope, this is the image of the microvilli. This one here. In the small intestine walls, these are the curvatures to increase the surface area. So in the lining, in the epithelial lining, so we can see here the cilia. So in detail, mura siya mga buhok-buhok. Nakita ni mo dira sa lining. Each individual cell is have villi kani. Microvilli. Gagmay pa jud siya. Micro man. Very small, microscopic villi. Okay, next we have the root hairs. So, what can you imagine when you say root hairs? Ay, sa buhok, ma'am, kada hair sa roots. No. These root hairs. Hair sa roots. Okay, are elongated hair-like structures that develop on the surface of the plant roots. Ganina ang microvilli sa animals, sa intestines. These root hairs are found in the roots of the plants. So what is the function of these root hairs? To increase the surface area of the root, enhancing its ability to absorb water and nutrients from the soil. The long, thin shape of root hairs allows them to penetrate the soil particles effectively. So the root hairs in plants are adaptations that primarily improve the plant's ability to absorb the water and the minerals from the soil. Okay? So that is the benefit of having modifications in the cells such as the root hairs and the microvilli. So let us now view a video about the cell modifications and what were the important features of cell modifications. Okay?
Good day! Welcome to our learning episode in General Biology 1. This subject is specialized for grade 12 Science, Technology, Engineering, and Mathematics STEM students. But of course, everyone who is willing and interested to learn is also welcome to join us. Today, we will talk about one of the interesting topics in cell biology, the cell modification. We will answer the following questions. What are the cell surface modifications? Where are the cell surface modifications located in an organism's body? How do cell surface modifications adapt to carry out specialized functions? Are you now ready to listen and learn? If yes, Make sure you have your paper and pen with you so that you can write what you will learn from this session. This is Teacher Jocelyn Handusay Barnizo of Nabunturan National Comprehensive High School, Integrated Senior High School, Nabunturan West District, welcoming you all to another exciting and fun-filled learning of science concepts through TV Eskwela. Before we will start our discussion, I have prepared 3 questions for a true or false pre-test. You will be given 10 seconds for each number to answer. Question number 1. The level of biological organization is arranged as follows: organs, organ systems, organism, tissues, cell. Time is up. The correct answer is false. Life here on Earth exhibits organization whether they are unicellular or multicellular organisms. The level of biological organization is as follows: cell, tissues, organs, organ systems, organism. The atom is the smallest unit of matter, followed by molecules. When molecules are grouped together, they ultimately form a cell. The cell is the basic unit of life. In multicellular organisms, like plants and animals, cells are grouped as tissues to perform a specific function. Different tissues can be grouped further and form organs. The organs form organ systems that make the function of the body more complex and efficient. Organ systems will then form the whole organisms. Question number 2. True or false? Epithelial tissues are commonly seen outside the body as coverings or as linings of organs and cavities. Time is up. The correct answer is true. Epithelial tissues are commonly seen outside the body as coverings or as linings of organs and cavities. Cells that make up this tissue can have distinct arrangements like simple cuboidal, simple columnar, simple squamous, stratified squamous. Question number 3. True or false? In many epithelial cells, the basal domain exhibits special structure surface modifications that carry out specific functions. Time is up. The correct answer is false. Epithelial cells exhibit distinct polarity. They have an apical domain, lateral domain, and basal domain. The apical domain exhibits special structural surface modifications that carry out specific functions for most organisms. Now, let's learn what are the surface modifications. Living organisms exhibit characteristics of life like they metabolize, grow and develop, respond to stimulus, reproduce, and lastly, adapt to their environment. Furthermore, living organisms are made up of cells that exhibit special structural surface modifications that adapt to carry out specific functions. The structural surface modifications include villi and microvilli, cilia, flagella, and root hairs. Do you want to know where the cell surface modifications located in an organism's body? How do cell surface modifications adapt to carry out specialized functions? Okay, let's get started. The first cell surface modifications are the villi and microvilli. Villi and microvilli are finger-like cytoplasmic projections on the apical surface of most epithelial cells. Here is the longitudinal section of a villus. It contains thin epithelium, 1 cell thick, to increase diffusion rate. The number and shape of the microvilli of a given cell type correlates with its absorptive capacity. In the small intestine, where it is most highly ordered, are even more uniform in appearance. Let's take a look on this video on how it functions. In our body, absorption of food mainly takes place in the small intestine. The inner wall of the small intestine is highly folded and has many small projections called villi. The structure of the villi is special, adapted for absorption. The villi are finger-like and large in number. This feature greatly increases the surface for absorption. Moreover, the epithelium of the villi is 1 cell thick. This provides a very short distance for diffusion of food molecules into the blood. The cell membrane of the epithelial cells has a large number of microvilli. This further increases the surface area for absorption. Each villus has a lymph vessel called lacteal. In the center, the lacteal is surrounded by a network of capillaries. This allows the absorbed food molecules to be carried away rapidly. Therefore, a steep concentration gradient of food molecules can be kept across the wall of the small intestine to increase the rate of diffusion of food molecules. See how the villi and microvilli functions for absorption of needed nutrients for our body. Another cell surface modification is the cilia. Cilia are hair-like luteal structures on the surface of some epithelial cells. They are surrounded by the cell membrane and contain a central pair of isolated microtubules, surrounded by 9 more pairs of microtubules. Inner core of cilia consists of a cytoskeleton called axoneme. In living cells, cilia have a rapid back and forth movement. But how does really it functions? Let's take a look on this video. In female mammals, the beating of cilia in the fallopian tube moves the egg cell from the ovary down to the uterus. Oh, too good to know about these specialized cells. But hey, there's more. In most ciliated epithelia, such as the respiratory tract, specifically on the trachea and bronchi, through these hair-like projections, it helps remove microbes and debris from the interior of the lungs. Let's take a look on this video. The air we breathe contains foreign matters such as dust, bacteria, and pollen. The hair-like cells called cilia, as well as goblet cell, can be found in the lining of the respiratory tract called the respiratory epithelium. Goblet cells secrete viscous mucus that forms a layer to trap foreign material. The wave-like action of the underlying cilia mobilizes the mucus layer from moving foreign matter and preventing it from entering the lungs. We can really appreciate here the function of cilia in terms of sweeping away bacteria and viruses that may enter our body through our nose. Indeed, this time, it is a must to wear a mask. Furthermore, cilia are also used for locomotion through liquids in organisms such as paramecium, a prokaryotic cell. Let's take a look on this video. Its whole body is covered with small hair-like filaments, cilia, which help in locomotion. The next cell surface modification that we will learn is the flagella. It is a tail-like projection that protrudes from the cell body of certain prokaryotic and eukaryotic cells and functions in locomotion. A eukaryotic flagellum is a bundle of 9 fused pairs of microtubule doublets surrounding 2 central single microtubules. Let's take a look on this video. Flagella serve for the propulsion of single cell, example, swimming of protozoa like the euglena, a prokaryotic cell. Another cell surface modification which is found in plants is the root hair. Root hairs are long, tubular-shaped outgrowth from root epidermal cells. It aids plants and nutrient acquisition where it absorbs water and nutrients from the soil. Furthermore, it is also for plants' anchorage to the soil and microbe interactions. There you have it! The cell surface modifications, its location, and adaptive functions. Let's have a little summary through this table and see how much you have learned on the following concepts. Alright, we have just finished our discussion. Now, it's time to assess how much you have learned through this 6-item multiple choice. Question number 1. What is the correct increasing order of the biological organization? A. Tissues, cells, organs, organ systems, organism. B. Organ systems, organs, organism, tissue, cells. C. Cells, tissues, organs, organ systems, organism. D. Organism, organ systems, cell, tissues, organs. Time is up. The correct answer is letter C. Question number 2. What cell modification that is hair-like and luteal in structure on the surface of respiratory epithelial cells sweeps foreign matter and preventing it from entering the lungs? A. Cilia. B. Flagella. C. Microvilli. D. Villi. Time is up. The correct answer is letter A. Question number 3. Which of the following statements best describes cell surface modifications? A. Specialized organelles of the cell that involve in cell division. B. Structures that adapt to its environment of most epithelial cells. C. Are made up of plasma membrane that functions for secretions of matters. D. Structures at the lining of epithelial cells that carry out specialized functions. Time is up. Correct answer is letter D. Question number 4. How do cilia differ from flagella in terms of structure? A. Cilia are located all through the prokaryotic cell. Flagella has only one protrusion. B. Cilia are for absorption and secretion. Flagella are for movement and locomotion. C. Inner core of cilia consists of cytoskeleton. Flagella contain actin filaments held together. D. Cilia are found on the linings of small intestines. Flagella are found on the linings of trachea. Time is up. Correct answer is letter A. Question number 5. In our body, how do villi and microvilli perform its function? A. It moves the egg cell from the ovary to the uterus during fertilization. B. It absorbs food molecules to be carried away rapidly in the small intestine. C. It sweeps mucus and tract particles inside the respiratory tract out from the body. D. It moves back and forth using its flagella inside the fallopian tube going to the uterus. Time is up. The correct answer is letter B. Question number 6. What is the function of root hair in the plant? A. It absorbs sunlight for photosynthesis. B. It aids in the absorption of nutrients from the soil. C. It removes dirt from the plant nutrients pathways. D. It moves back and forth to give way for air to enter. Time is up. The correct answer is letter B. Have you get it right, my dear learners? If yes, Congratulations! Before we will end, let me share to you this simple quote from an American teacher, Frederick Lenz, saying, "You don't need a special test. Every task is special." That is also very true with the cell surface modifications. Their tests are special so as with the other parts of an organism's body. So, have you learned something today? Thank you for your listening ears and cooperation. For more science learning, tune in again to our next episode. Again, this is your teacher Jocelyn Handusay Parmiso saying, dito sa TV Eskwela, sa pag-aaral, sama-sama. Bye-bye!
Thank you.
Good day. Welcome to our learning.
Yes, thank you, Ma'am Jocelyn. So that is about our cell modifications. Sino nakakuha ng perfect score? Okay. So let us now proceed with the next topic, which is the cell cycle. A cycle. So when we say a cycle, like bicycle, tricycle, so what does a cycle means? A cycle refers to a series of events, a sequence of events or actions or processes that repeat in a predictable pattern or sequence. Okay? So in science, cycle refers to a circular or repeating process or series of events such as cell cycle, carbon cycle, ano pa ba? Nitrogen cycle, water cycle. So, yun. So, these cycles involve a sequence of stages that are essential for the functioning of the system or organism. So, the cell cycle is the series of events that occur in a cell from one division to the next. Okay? So, here, I prepared an illustration of the cell division. So, a cell cycle is a highly regulated cycle or events that occur in the eukaryotic cells. So, where the cells divides into two daughter cells. It is a process from the formation of a new cell through cell growth. Wait lang. Agay. Okay. So, the DNA and the DNA will also replicate following the cell division to the formation of two identical daughter cells. Okay? So, the cell cycle is important. Why? It is important for the growth, development, repair, and maintenance of the multicellular organisms. So, the cell cycle consists of different phases. So, what are these phases? So, we have the interphase. Okay, this is the interphase, the whole of it. So, interphase is the longest phase and it is divided into phases. The gap 1, the synthesis, and the gap 2 phase. While this small portion is what we call the mitosis. So, mitosis is the phase of the cell cycle in which the nucleus of the cell divides. This mitosis is divided into the following phases: the prophase, metaphase, anaphase, telophase, and the cytokinesis. Okay? These are the phases. of the cell cycle. The interphase composed of G1, S, and G2, and mitosis composed of prophase, metaphase, anaphase, telophase, and cytokinesis. Okay? And here is the checkpoints. Diba? Murag LTO na ay checkpoint. Na checkpoint ang ato ang kuan. Cell cycle. Nga naman, para madakpan ang mga kolorum. Okay? Para ma-check kung naabay mga illegal na mga nahitabo or mga wrong processes so that dili na siya maka-proceed. Ma-stop na niya. Okay, once katong mali nga division ang nahitabo, then wala na check sa ato ang checkpoints, and then mag-continue ang process. So ang na duplicate nga daughter cells kay unsa man naan na siya mali ato. Okay? So, let us proceed with the interphase. So, what happens during the interphase? So, we said that interphase is the longest event in the cell cycle. Preparation. Wala nag-divide during interphase, ha? Nagpahuway ang cell. Pero wala siya nagpahuway nga, wala siya gibuhat. Wala lang siya nag-divide, pero nag-prepare niya ang cell for cell division. So, during the interphase, the cell cycle have made important events. And it prepares the cell for cell division. The interphase is the longest phase in the cell cycle, divided into subphases. The G1 or gap 1 phase, the S or the synthesis phase, S stands for synthesis, and G2 meaning gap 2 phase. Okay? So here, in the next slide, is the description on what happened in the first gap. So, during gap 1, there are also important events that happen or occur during the gap 1, such as the cell prepares for DNA replication, and future cell division. So, the focus of G1 is to what? To prepare for DNA replication. Okay? So, the G1 focus on cell growth. The G1 is characterized by significant cell growth. Grow ang cell sa G1. So, during this phase, the cell accumulates the necessary resources and cellular components to support DNA replication and future cell division. This growth involves the synthesis of proteins, lipids, and organelles such as mitochondria and endoplasmic reticulum. So, magpadako sa ang cell. Then, also, the cell carries out its normal function and activities during G1. Normal nga iyang function. Activities depend on the specific type of the cell and the function of the cell within the organism. For example, if the cell is a liver cell, it continues to perform its metabolic function. And if it is a skin cell, it maintains its role in forming outer protective layer of the skin. So in G1, the cell will grow and it will do its normal function. Kung sa iyang trabaho, kung sa siya nga cell type, mo to iyang himuon. Normal niya nga trabaho. Ug magpadako siya. Okay? Next is the cell cycle control system. A set of molecules and that triggers and coordinates important events in the cell. This control system is responsible for the regulation of a certain checkpoints in the cell. So, naatay LTO sa ato ang cell. Naatay control system. It is a highly regulated network of proteins, molecules, and checkpoints that governs and coordinate the progression of eukaryotic cell through the various phases of the cell cycle. So, what's the function of this control system? To ensure that each phases of the cell cycle is completed accurately, and the cell only proceeds to the next phase when certain conditions are met. So if the conditions are not met, the cell will not proceed to the next level of cell cycle. Okay? So the cell cycle control system plays a crucial role in maintaining genomic stability and preventing uncontrolled cell division, which can lead to diseases like cancer. So, ang ato yung lawas, naadjud siya kuan system, naatay cell cycle control system that prevents uncontrolled cell division, that prevents certain data nga wala or mali sa DNA nga dili siya ma-prevent jud para mag-occur. Pero question, ngano na manggihapoy cancer, ma'am? Ngano na manggihapoy mga disorders? Kay tungod wala ni gana or wala nagtrabaho og tarong ang mga LTO sa imong lawas. Nanguan sila. Wala sila nag-duty. Charla. Bitaw class, there is something wrong with your cell cycle control system. They are not doing their function. Because when they do their function, they can check the uncontrolled cell division. They can check if merong mga inconsistencies and mga disorders. And if they detect one, they will stop cell replication. They will stop the cell cycle unless all the standards or all the conditions are met. Okay? So nga naman gihapon nga naa may cancers because we are exposed to carcinogens. We are exposed to stress. And if that is the case, we lower the level of our immune response, of our immune system. We weaken our cell cycle control system. So if our cell cycle control system is weak, it cannot function well. It cannot function properly. And if that happened, uncontrolled cell division will occur. Cancer will occur. Okay? So, what are these checkpoints? Tanaw ta ha, these are the checkpoints. The first checkpoint is through the gap 1. Gap 1 checkpoints, the cell continually monitors its internal and external environment. So, regulatory checkpoints in the cell cycle assess factors such as the integrity of the DNA, the presence of growth factors, and the overall readiness of the cell to proceed to the next phase of the cell cycle. If the cell is not ready, it cannot proceed to the next cycle. Decision to divide or not to divide. So, if the G1 phase determines the cell's fate, if it can divide or it cannot. So, at the G1 checkpoint, the cell assessed whether it should proceed to the cell cycle, including DNA replication and division. Factors that influence its decision include the health of the cell and the availability of the nutrients and the resources. Okay? And it also signals, it also follows the signals from neighboring cells. Okay, so if the cell receives the appropriate signal and conditions are favorable, it will continue through the cell cycle and enters the S phase or the synthesis phase where the DNA replication occurs. However, if the conditions are unfavorable or if there is a DNA damage that cannot be repaired, the cell may exit the cell cycle and enter a state called GO or quiescence, where it remains a non-dividing but functional state until conditions become favorable. So, dili sa siya pa pa-divide. Dito ra siya, patrabahoon ra siya sa mga normal nga trabaho niya. Pero dili siya pwede i-divide kay naa siya'y damage iyahang DNA. Okay? So, during the gap 1, the cell undergoes growth, carries out its normal function, and evaluates whether it is suitable to proceed to the next cycle. It is a critical stage that helps to ensure the cell division occurs under appropriate conditions and the genetic material is replicated accurately. So we have here stimulating protein and inhibiting protein. So what are the functions of this one? So these are the what? The traffic signals. So these are the signals. These are the proteins. So this CDK cyclin complex is like a traffic light. So think of a traffic light at a center at an intersection. So mo na siya mga traffic light. Because this cyclin proteins act like a traffic light and the CDK proteins are like controller that can turn the light green. When the light is green, activated CDK cyclin complex, the cell can proceed to the next phase. Okay? So, daghan pa ni mga proteins nga mag-act as the cell cycle control system, mga police, mga traffic enforcer. How about daghan pa? P53 protein is like a police patrolling the cell. So if the P53 spots any trouble like DNA damage, it can help the cell cycle by turning the light red, inhibiting the CDK cyclin complex. It ensures the cell is safe and the DNA is intact before allowing it to continue. How about RB? RB protein is like a gate. Lock in a gate. Where it's locked or unphosphorylated, it prevents the cell from moving forward. But when certain keys phosphorylates from activated CDK cyclin complex, it unlocks. The gate opens and the cell is allowed to progress. Phosphorylates nga, nadungagan siya mga chemicals. Okay? Next. Mura man to growth factors. So what's the function? Mura po siya traffic signals, traffic light pareha ni. So growth factors are like signals from other cars at the intersection. So when the signals are received, they can influence the traffic light to turn green, allowing the cell to move forwards. So in those terms, the proteins and the inhibiting proteins, they are the traffic light that turn green to let the cell proceed or red to stop it. Okay, so the P53 watches for trouble and can stop the cell if needed. The RB proteins are the lock and key, controls whether the gate to the next phase is open or closed. And the growth factors are also plays a role in the decision making to ensure the cell is safe before continuing. Next, we have the S phase. So the S phase is the preparatory phase for the cell cycle. So this is where the DNA replication occurs. So what happens during the S phase? Takes a lot of time because this the complexity of replicating the genetic material. This is where DNA replication occurs. So think the cell as a library that holds all the genetic information. During the S phase, the cell makes exact copies of all these books. These copies are like photocopies of the original books. So it replicates, it copies the DNA of the cell. So unzipping of the DNA. So unzipping. I-open ang zipper. So imagine that the pages of each book are stuck together like a zipper. So the enzymes in the cell carefully unzips or unlocks the pages so that they can be copied. Special workers in the cell called DNA copiers read the text of the pages and make new pages that are identical to the ones they read. So every book or every DNA, you end up with 2 identical copies. So marang nag-photocopies. The cell is very careful, like a proofreader, and checks to make sure that there are no mistakes in the copied pages. So if there are errors, they will try to fix them. So, while all this copying is ongoing, the cell also gets ready for the cell division. It prepares extra materials and makes sure everything is ready for the next step in the cell cycle. So once the S phase is finished, the cell has 2 complete sets of instructions. Okay? For the building and operating of the cell. So the cell is now ready for the cell cycle and divide into 2 new daughter cells. So we also have this pagkatapos ng S phase. Proceed na naman tayo sa G2 or gap 2. So, what happened during gap 2? The cell is ready to divide. So, what happens? So, imagine the cell as a factory. During the G2 phase, it's like a factory is doing some final preparations before the big project. No? So, the factory or the cell checks its blueprints, and that is the DNA. So, it makes sure that the copies of DNA made during the S phase are accurate and complete. If there are any mistakes or damage in the DNA, the cell sends its workers to fix them. They make sure the instructions are correct and perfect. So, the cell double checks that if it has all the tools and materials it needs for the upcoming cell division. If it is good, the factory manager or the cell control system gives go signal to the special complex proteins like the cyclin CDK, so ready for cell division. Okay? So, the G2 phase is the final checkpoint, the final preparation of the cell for the cell division. It ensures that everything is in order and ready for the cell division to run smoothly. So, we have the checkpoints in the G2 phase. This ensures that the DNA had been accurately replicated without mistake or damage. If it detects a problem, the cell will either complete the replication checks or stop. So these are the proteins responsible for the checkpoint, the cyclins and the CDK and the P53 proteins. Okay? Yes. We will view a video about this cell cycle.
In this lesson, we'll be looking at the cell cycle. This is the lifespan of a eukaryotic somatic cell. A somatic cell is any cell in the body of an organism, except for sex cells, such as sperm and egg cells. The cell cycle describes the sequence of cell growth and division. A cell spends most of its life in a state called interphase. Interphase has 3 phases: the G1, S, and G2 phases. Interphase is followed by cell division, which has 1 phase, the M phase. Together, these 4 phases make up the entire cell cycle. G1 of interphase is sometimes called growth 1 or gap phase 1. In G1, a cell is busy growing and carrying out whatever function it's supposed to do. Note that some cells, such as muscle and nerve cells, exit the cell cycle after G1 because they do not divide again. A cell enters the S phase after it grows to the point where it's no longer able to function well and needs to divide. The S stands for synthesis, which means to make, because a copy of DNA is being made during this phase. Once DNA replication is complete, the cell enters the shortest and last part of interphase called G2, also known as growth 2 or gap phase 2. Right now, it's enough to know that further preparations for cell division take place in the G2 phase. Now that interphase is over, the cell is ready for cell division, which happens in the M phase. The M phase has two events. The main one is mitosis, which is division of the cell's nucleus, followed by cytokinesis, a division of the cytoplasm. So at the end of M phase, you have two daughter cells identical to each other and identical to the original cell. Let's review. The cell cycle describes the life cycle of an individual cell. It has 4 phases, 3 in interphase and 1 for cell division. Most cell growth and function happen during G1. The cell enters the S phase when it needs to divide. In this phase, the cell replicates its DNA. Replication just means the cell makes a copy of its DNA. In G2, the cell undergoes further preparations for cell division. Finally, we have cell division in the M phase. The M phase consists of mitosis, which is nuclear division, and cytokinesis, or division of the cytoplasm. We'll explore the details of mitosis and cytokinesis separately.